Hi everyone and welcome to week 4, and in this week we're going to look at DNA, we're going to look at protein synthesis, okay, and this is often called translation, okay. So remember the messenger RNA we made in transcription? What we're going to do in translation is we're going to take that message and we're going to translate it to a protein. So if you look at this picture, this is a picture of hemoglobin, and this is a protein that we can make through this entire process that we've talked about in this course. So it started with the DNA. We duplicated the DNA and we made new DNA. After that, we looked at messenger RNA and we made messages of the DNA. And what we're going to do is we're going to read that messenger RNA, and that messenger RNA is going to tell us and make a protein just like hemoglobin. And hemoglobin is found in your blood. And it's very important that hemoglobin is made correctly. And one way we make sure it's made correctly is by translation. Alright, so remember, we made mRNA in the last week. Okay, that was called transcription. So we took DNA, we read it with RNA polymerase, and RNA polymerase made a messenger RNA from that. So let me get you to think about something for a second. When all of this is happening, we need to make sure we understand where it's happening. So if we're thinking about making mRNA, where is the DNA located in a cell? The DNA is located in the nucleus of the cell. So to have mRNA made, that has to occur in the nucleus, where the DNA is. Okay? Now, the next process we're going to talk about, translation, does not happen in the nucleus. It happens outside the nucleus. Okay, so there's a lot of different terms that we're going to look at in this slide, and I want you to go ahead and write these down in your notes, and we're going to see if we can identify these. You can go ahead and write down the definitions for a few because we've already talked about them. Number 2 is mRNA, and that is a messenger RNA that carries a message to make a protein. A codon is a 3-nucleotide sequence found on the mRNA, and everything else we haven't quite talked about yet, so we'll get to those in a minute. Okay, so this is a lot, this is something new, okay, and I want you to look at it very carefully. So what we have down here is we have mRNA, okay? During translation, mRNA is read by an organelle called a ribosome. Okay, and we've talked about ribosomes before. Ribosomes make proteins, and we're going to show you how that happens. Okay, so in order to make a protein, we have to have mRNA, which was what we looked at last week. So here's our mRNA. So what's going to happen is we're going to have a lot of different things that go on, all right, and we're going to kind of go through it a couple times so you can understand what's happening. All right, there are 3 major sites inside of a ribosome. Okay, the ribosome is represented by this blue figure. The ribosome has a large subunit and a small subunit. The large subunit is located, it kind of attaches to the top of the mRNA, and the small subunit attaches to the bottom. So it's kind of like a clamp to hold on to the mRNA. Okay, so the first section where the RNA looks at the DNA or the mRNA, the ribosome starts at the A site. Okay, then it moves to the P site, then it moves to the E site. Okay, so let me see if I can kind of show you how this works. So here is our RNA, and it's traveling in this direction. I'm sorry, it would travel this way. Okay, so it goes to the A site, it reads those 3 codons, then they move right along, it reads those 3 codons, it moves right along, and it moves those 3, and it continues like so. Okay, so it starts this way and it goes that way. Okay, so that's just kind of a view of what's happening. Okay, so remember we said codons were really important. All right, this is where they are very important. What the ribosome is reading is it's reading the codons on the mRNA. When it reads a certain sequence of codon, it brings in something called a tRNA. tRNA, the T in tRNA stands for transfer. Okay, transfer RNA. What is that RNA molecule transferring? Well, it's transferring these little beads. It looks like beads on a string. What are those beads? What does those little circles represent? Those circles represent an amino acid. And if you remember from our previous lessons, amino acids are the building blocks for proteins. So this is where we make a protein. Okay? So this is a very specific tRNA, and what it's doing is it is reading, or it is matching up with this codon, as you can see. This G matches up with that C, this G matches up with that C, and this C matches up with that G. So every tRNA is a little bit different. This tRNA has a specific amino acid there. Okay? So when it reads this codon, it attaches the correct amino acid. Okay? So what is this part of the tRNA called? Well, if this is a codon, this has to be an anticodon. So anticodons are located on tRNAs that are complementary to codons found on mRNAs, which allows them to make the correct protein. Okay? So as we can see, the RNA molecule is traveling from left to right, and it is going to continue to move through this ribosome. Okay? So as it moves through the ribosome, it goes through those different sites that we looked at in this slide. The A site, the P site, and the E site. The A site is where it goes in first, then the P site, then the E site. The way I remember these is I remember that A is kind of where the tRNA is accepted. P site is where the protein is made, and the E site is where it exits. Okay, so accepted, protein, exit. That's kind of how I remember A site, P site, E site. Okay, so now, as you can see, as the molecule, the RNA molecule moves along, this E site is where the tRNA exits. Okay, so as it continues, this chain will continue to get longer and longer and longer, and finally, when it's done, we will make a complete protein. Okay? Now, there are certain sequences within the mRNA that make the process of translation stop, and these are called stop codons. So one of the stop codons is UUA. And you're going to need to know that for your test. You're going to need to know this stop codon. Okay? So what does it do is it stops translation. That means that the protein is made and the ribosome doesn't need to read any more nucleotides. So it reads UUA and then it stops, and then we've made the protein that we wanted. Alright, you've made it to the end. Please go back and review this video and make sure you understand the process. If you have any questions, just email me and I'll be glad to help.